This is John. I work for Bernard Jasmin at the University of Ottawa. One of the things we do when we're investigating muscular dystrophy is we work with cells that are in culture. So I have a dish here of cells in culture. You can't see them, unfortunately. But what we do with the cells in culture is we try different types of drugs and treatments on the cells prior to using them in the animal. So this is basically a biosafety cabinet, and this is a cell culture practice. Thank you.
I'm a research associate in Dr. Bernard Jasmin's lab, and today I'm going to show you some of the mouse models that we use to study muscular dystrophy in our lab. So here we have our rack, which contains cages that have all of the mice. We have three different mouse lines that we study. So there's wild type or control mouse, and then we have a dystrophin deficient mouse model called MDX, as well as a dystrophin and a utrophin double knockout mouse called the double knockouts. And we house them here, and if we want to use mice for our experiments, we set up breeding colonies and use their offspring to study muscular dystrophy. So when we need to collect samples or change cages or separate mice, we use a fume hood here, or laminar flow hood, and we open up the cage that the mouse is kept in, and as you can see here, the mice that we use are black in color, and they have ear clips to indicate their mouse number so that we can identify which mouse is what. And yeah, that's what our mice look like, and this is how we study our muscular dystrophy mice.
Hi, my name is Christine Pelado. I'm a PhD student in Bernard Jasmin's laboratory. My research has been focusing on Duchenne muscular dystrophy. As you may see here, I'm using a specialized microscope called the ELO. And here we're looking at histology sections of the MDX mouse muscle, which is the mouse that has muscular dystrophy. And as you may see here, we can see at all different aspects, and this permits us to study the muscle in very specific details.
Hi, this is Bernard Jasmin. I'm the Dean in the Faculty of Medicine at the University of Ottawa. I just want to say a big thank you to Jesse's Journey and all the volunteers and the donors and all supporters. Through the funds of Jesse's Journey, we've been able to carry out our research program dealing with the regulation of utrophin in skeletal muscle. The main idea here is to try to upregulate the levels of this protein, which could substitute itself for the lack of dystrophin. Our work in recent years has resulted in the identification of potential drugs that could be of benefit to patients with DMD, and we look forward to continuing our interactions and collaboration with Jesse's Journey as we will try to bring those drugs to the clinic. Thank you again, everyone, and we look forward to more interactions with all of you.